All right, let's try this again, y'all. Keep your fingers crossed. Okay. Oh, yay. Oh, we're back in business. Yes. Okay, now let me find. Nope, that was not what I wanted to do. That's okay. We'll just kind of click through them. It's okay. Okay. Woohoo. All right. Whoa, yes. All right. So reducing sugars. When we talk about reducing sugars, basically what you're looking for, do you know oxidation reduction? Oil rig, right? You remember this? So oxidation is loss of what? Electrons. Rig is gain of electrons. But what else is it when we talk about organic? Oxidation is also gain of oxygen, loss of hydrogen. Reduction is gain of the opposite, hydrogen, and then loss of oxygen. Y'all remember that? Yes? Okay. So what happens when we go from D-glucose to D-gluconate? What changes? We gained a oxygen, we lost a hydrogen. Okay, so what did we do? We did oxidation of our sugar. And so in the process, you can do this test. And it's called a Benedict's test. And so you have a test tube and you have the Cu2+ ion, which is blue. And then when the sugar becomes oxidized, the copper becomes reduced and the copper actually precipitates red. And so you can actually test someone's sugar levels using urine and then being able to see if their glucose levels are too high. So this is kind of one of the early ways that they tested people's blood sugar, and it was really mostly urine. But they actually do a new test now, and we'll talk about it a little bit. But the new test that they do now is that they take your blood sample, so you have glucose in it, and glucose is a reducing sugar, which we just saw. And so you convert that to gluconate, G-gluconate, G-N-A-T-E. There's no G in it. Okay, from glucose to gluconate, right? And so you do this with an enzyme. You do this with glucose oxidase. But what happens in this process with glucose oxidase is that you have to do this in the presence of O2. And when you do this in the presence of O2, you make H2O2, hydrogen peroxide, right? And then we can take advantage of the presence of this hydrogen peroxide. And this hydrogen peroxide will catalyze basically a dye changing color from one color to color two, right? Color two, with peroxidase. Why does it do that? Let's do this. So the dye changes color through peroxidase. Okay. Oh, and I wanted to put my chat up. I'm sorry. Sorry, y'all. I'm not ignoring you. And I hope it's recording again. Yes, it is. Okay, yay. So this is a much better, a much more sensitive test, right? Than these early tests. Even though it was good, it did give us some information. But really, we're not normally looking at monosaccharides inside of our body. We're usually at least looking at disaccharides, right? So how do you link monosaccharides? With a glycosidic bond, right? And what links the two is an oxygen between the two. So we call it an O-glycosidic bond, right? And it forms through a condensation reaction. So if you're going to make a glycosidic bond, you're going to start out with two sugars. Each sugar has an OH attached to it. So I'm just going to represent the sugar as an R, right? And then I have another sugar that has an OH. So what happens in a condensation reaction? I'm going to take OH and H, and I'm going to make what? Water. And then what's my resulting I get R, O, and then it's bonded to the other R, right? And that's how I get a disaccharide. So there are some common disaccharides that you need to know. You need to know lactose. Let's see if I can clear some of this. So that's just your general reaction. But lactose. So anybody know what the components of lactose are? Because it's usually not two of the same monosaccharide. Usually it's multiple, like two different kinds. Like lactose is galactose and anybody new? Glucose? Glucose, good. And it's linked. These two monosaccharides are linked inside of a glycosidic bond. So in this glycosidic bond, it is a beta 1,4 bond. And I'm going to show you some of these bonds in a second. Sucrose. I bet you everybody has some sucrose at their house. Not everybody necessarily has lactose, but sucrose definitely. Where's sucrose? It's like table sugar, right? Where's lactose? In your milk, right? In your milk. So some people might not drink milk, but sucrose is glucose and fructose. And it is in a alpha 1 beta 2. Okay, why did the nomenclature change? Why are we a beta 1,4 and an alpha 1 beta 2? Anybody have any ideas? Because the anomeric carbon is both participating in the glycosidic bond. So if you're listing 2, so both, that means that both anomeric, M-E-R-I, anomeric carbons, hello, participate in the bond. Glucose is alpha, fructose is beta. Yes, yes. Both anomeric carbons in the sugars participate in glycosidic bond. Our last one here, this is one you probably are not familiar with. This one is actually found in insects, but it's kind of cool because it's a glucose-glucose. It's a glucose-glucose disaccharide. So this is an alpha 1 bond. So let me show you what some of these bonds look like. All right. So this is lactose. Lactose, we said, was galactose and glucose, and it's in a beta 1,4 glycosidic bond. Where are my 2 anomeric carbons? Carbon connected to 2 oxygens. So I'm going to put a dot right there. That's an anomeric carbon. It's connected to 2 oxygens. Now, where's my other anomeric carbon? On the other side. So as long as I don't have another carbon attached to it, this is carbon number 1, right? So that's where my beta 1 comes from, right? So it's in carbon 1. Now, where's the 4 come from? Well, if this is carbon 1, 2, 3, 4, do you see how it's 1 and 4 participating in the glycosidic bond? Yeah. Okay, and then beta is telling me that it's in a cis orientation. So you could actually take this and move it up so that you could see, but do you see how it's pointing up here? That's telling you that it's above the ring, just like CH2OH. I don't particularly care for that drawing, but it's okay. Look at sucrose. If we have an alpha 1, beta 2. So here's my anomeric carbon, that's carbon 1. And then where's my other anomeric carbon? Right here, right? Why isn't that carbon 1? What's attached to my anomeric carbon that's outside of the ring? A carbon. See my CH2OH? That's my carbon 1. This is my carbon 2. Got to be careful of that. Be careful of that. Okay. And then So this one, I'm looking at my CH2OH. Do you see how they're trans? They're opposites. So trans is alpha. That's alpha 1. And then if we look here, right, CH2, not CH2OH. Yeah, CH2OH. This one is beta. So when we look at this, what do you notice that's different between lactose and sucrose? Do you see how sucrose, we have both anomeric carbons that are participating in the glycosidic bond? That means that this sugar is non-reducing. Here, only one of the anomeric carbons participates in the glycosidic bond. So this is a reducing end. That means that this carbon is still very reactive, and so can participate in oxidation reduction. So look at our last one, Triolos. Where are our anomeric carbons? One here and one here, right? So this is carbon 1. I don't have any other carbons attached to it, right? Do I have any other carbons attached to this one? No, so this is a 1. And then they are both alpha. Why did you number the CH? Okay, so William, here, this one, you have to do this because this means, what kind of a sugar is it? This is the carbon that was in the double bond with the oxygen, right? And so if you had a carbon above it in the Fisher diagram, so if I go back real quick, I want to go back here, right? It was like this. It was like this one, where this is the anomeric carbon. This is what becomes the anomeric carbon. So it's technically carbon 2 that's the anomeric carbon, and then this carbon is outside of the ring. So we have to number this carbon as number 1, and then our anomeric carbon becomes number 2. Whereas here, this was the carbon that was inside, that is the anomeric carbon, but it's carbon 1 because there's nothing else attached to it. So you're talking about either an aldehyde or a ketone.
So whenever you have fructose system, 2 carbons outside of the ring, which one do you use to determine whether it's beta or alpha?
It's the one that's bound to the oxygen that's bound to the anomeric carbon. Yes, it's a good question. Okay, so let's see, where were we? Okay, we were here, right? Okay, and we said alpha 1, alpha 1. We okay on that? Yeah? Okay. So if you notice there are 2 CH2 groups, then you number those first and last. Yeah, so you find your anomeric carbon first. If you find your anomeric carbon first, then you know where your numbering goes. Always go and find your anomeric carbon. Right? And then you're looking at the CH2OH that's not bound to the anomeric carbon, but is bound to the oxygen to see if it is alpha or beta. Can't believe y'all didn't get this in organic. Okay. So how do you determine if it's alpha or beta? So find your anomeric carbon. That is your first step for everything. I'm going to find my anomeric carbon. Then I need to number it, right? If there's no other carbon attached to my anomeric carbon, it starts at number 1. If I have another carbon attached to my anomeric carbon, which I don't have on, oh, I do have right here. So here's my anomeric carbon. I have something attached to it. So then I number it this way, right? You're not going to use this to determine if this is alpha or beta. So carbon 1 is not it. It's going to be the farthest carbon. So this is 1, 2, 3, 4, 5, 6. So carbon 6 is going to, and it might not be 6, but it's the biggest number, right? So in this case, right, it was 1, 2, 3, 4, 5, 6. So it was carbon 6 and the hydroxyl on the anomeric carbon. On fructose. So on fructose, we now have our glycosidic bond. So we're looking here, and this one's going down, and then we go find our carbon 6, and this one is going down. So it's in the cis orientation, so it is beta. Here, I found my anomeric carbon. I go find carbon number 6. I compare carbon number 6 to my glycosidic bond. I am what? I am opposite, so I am trans or alpha. Here, I found my anomeric carbon, right? I go around, find carbon 6, look at my bond. It's pointing up. I know that's crazy, but it's pointing up. So this is on the same side. So this is cis or beta. Right here, I find my anomeric carbon. There's my carbon 6. So I look at my bond. My bond is going down. My carbon 6 is going up. So I'm beta, but I'm not going to list it because it's not an anomeric carbon participating. But it is beta. But it's not an anomeric carbon that participates, so we don't list it. That's how we know it's not the anomeric carbon participating in the glycosidic bond. Yeah? Okay. All right. So last little bit. So basically you're going to eat things, right? And your body's going to break them down. For example, maltose. Anybody know what maltose comes from? So you eat starch, you eat plant starch, right? And then you have in your saliva alpha amylase. Alpha amylase breaks down starch into maltose, right? So now you have maltose hanging out. Well, what's going to happen? You're going to cleave maltose into glucose and glucose. That's great. You can shuttle that straight into glycolysis. Sucrose, table sugar. What are you going to get? Glucose and, fructose. So guess what's going to happen? In order to get this molecule of fructose in glycolysis, you're going to have to make some changes to it. So you have to remember that not every sugar that you break down can directly go into glycolysis, right? Lactose is going to break down into glucose and galactose. You're going to have to modify galactose to make glucose to get it into glycolysis. And we'll talk about how we do that, right? The other thing to kind of remember, right, is all this stuff, right? Once you get it to glucose, you're going to go through glycolysis. What happens after glycolysis? SIS. You're going to make what in glycolysis? What's your end product? You should pyruvate. Pyruvate. So you have pyruvate. You want to go through the citrate cycle. What are you going to turn pyruvate into? Like these are the big ones. You should probably have gotten in like biology 1. Acetyl-CoA. ACETY. Acetyl-CoA. Yeah. So once you have acetyl-CoA, you can take acetyl-CoA and you can put it into, maybe you heard it as Krebs cycle, right? Same thing. Okay. So the other thing that can happen is that you can eat other things that aren't sugars, can't you? Right? And we can use them for energy. How does that work? Well, what if you eat a triglyceride? Many of us eat triglycerides, right? We go through some enzymatic reactions with lipases. And what do you make? Fatty acids. What can we turn fatty acids into? They're not going to enter glycolysis. They're actually going to get converted to acetyl-CoA. And then we can pick up with that pathway. So you'll see a lot of things feed into and out of these big, diverse pathways. And I think my chat's too far down. Okay. Since they're not exactly the same because they don't have the same linkages. Adela. Okay. All right, and I'm going to talk about lactase and lactose intolerance in the video that I already did. So we'll talk about that now. All right, so glycolysis, loving glycolysis. All right, so we go through glycolysis. What are we trying to prepare for by going through glycolysis? Like, what's our next step? We do glycolysis, and then we just said, Krebs or the, oh my gosh, I'm too far up close to the board. Citrate cycle. What happens after the citrate cycle? That's going to be our last chapter. These are our 3 chapters: glycolysis, citrate cycle, and oxidative phosphorylation. Right? So we got to get our sugar ready for the citrate cycle, or the Krebs cycle, whatever you want to call it, right? So our whole goal is to take glycolysis and to split it into 2 molecules of pyruvate. That's our end goal. But what's our big goal for glycolysis, citrate, oxidative phosphorylation? Do you remember writing the equation for cellular respiration in class, right? What do you got? I know you can do it. C6H12O6 plus 6O2 in a reversible reaction, right? So what? 6 CO2 plus 6 H2O. Yes, so we're eventually going to end up with just carbon dioxide and water, right? And so the very beginning of this is just converting from a 6-carbon sugar into a 3-carbon, right? And can we harvest some energy from that? And so basically you have 10 enzymatic reactions that we separate into 2 stages. One stage we call the investment stage. So when you invest in something, what do you have to do? You have to spend money, right? This is investment. You spend money to get money, right? You've heard that. So we're going to spend a little money in stage 1. And what kind of money do we spend in the cell? ATP. So we're going to spend some of that money, some of that ATP. We're going to actually invest. Anybody know how much we're going to invest? 2 ATP. And then we're going to get a payout in stage 2. What's our payout in stage 2? 4 ATP for a net of plus 2 ATP. So glycolysis is this core metabolic pathway, and we know it's a core metabolic pathway. We know it's super insanely important because it is conserved across all different organisms, right? So we think that it's a really, really ancient pathway that's been here for a really long time. And so this is how we can generate ATP even if we're under anaerobic conditions. What are anaerobic conditions? What? No oxygen. We don't have oxygen, right? And so you have some organisms that can do that, right? But you also have some cells in your body that do that. Do you know what they are? If you don't have a mitochondria, you can't do aerobic. You have to do anaerobic. So which cells don't have a mitochondria? No, our cells. There are some bacteria that undergo anaerobic, but red blood cells. Red blood cells are like sacks of hemoglobin. They don't even have a nucleus anymore, much less a mitochondria, right? But they have to function. They have to generate ATP somehow. And so this is what happens inside of red blood cells so that they can get energy, right? And then the other big thing is that metabolites are precursors, right, for all kinds of interdependent pathways, right? Remember we saw that big, insane maze of metabolic pathways, right? So everything's really interconnected. And so some precursors are from other pathways, and sometimes they get shuttled out in order to function in other pathways. So it's all kinds of interchanging of metabolites. I don't know another word to say of it, right? But okay, so what do we do in glycolysis? Here's our overall reaction for glycolysis. We're going to take glucose. We need NAD+. We need ADP, and we need 2 inorganic phosphates. And what are we going to make? This is our net reaction, not step by step, but this is our net reaction. We're going to make 2 pyruvate. We're going to make 2 NADH, which are going to be important for later. We're going to make 2 hydrogen ions, 2 ATP, and some water. Okay, so make sure that you can recognize the difference between the overall reaction for cellular respiration versus glycolysis alone, right? Okay, so your book has this really cool little animation, and oh, I loaded it before I closed the computer. Oh, dang it. I really did load it before. So I'm going to post it for y'all. But it's a really nice animation that shows the different stages of glycolysis, where you have this initial investment phase, and then you have a production phase, where you make the 2 ATPs that you invested, plus you make 2 more ATPs, okay? And so what you have to remember is that once you get out of the investment phase, the last part of the investment phase is where we break our glucose into 2 3-carbon molecules. They're not pyruvate yet. They have to go through some changes to get to pyruvate. But you do have, so this is a 6-carbon, this is a 3-carbon, this is a 3-carbon. So when we look at the reactions that go on the side here, what you're going to see, I'm only going to show you one, and you just have to remember it's multiplied by 2. Okay? So the production phase is split into 2 parts. The earnings, this part, the green part at the bottom, yes. So basically, you have your glucose, you split it into 2 molecules at this point. They're not pyruvate yet, even though pyruvate is a 3-carbon molecule. But you have to do some modifications to it. And so we're going to look at one of them, but you got to remember that 2 molecules are going through the same pathway at the same time. I'll show you. Okay. So when we talk about what happens in glycolysis, you got to remember where you are in the cell. Where does glycolysis occur? Where are we? Here's glycolysis. Where are we? In the cytoplasm, in the cytosol, right? And so you got to think that once we go from glucose to pyruvate, now our pyruvate can take one of 3 different pathways, right? It can go into the mitochondria, and it can go into the mitochondrial matrix. Our pyruvate can be oxidized, and we can turn it into carbon dioxide and water, right? That's what we've kind of been talking about. But under anaerobic conditions, there are 2 different pathways that can happen. You can turn it into lactate. That's what happens in our muscles. Or you can turn it into ethanol and carbon dioxide, and you can make beer with yeast, right? And I actually tell you how you do that in the last section. You can also store it as fatty acids, right? If you have too much glucose, what you can actually do is you can convert that into a storage molecule, right? And so you have to remember that there's lots of interconnections of all kinds of different pathways, and I don't want you to lose sight of that, right? All right, so here's kind of our glycolysis overview, and we're going to take it step by step and go through each and see why each step is important. But I thought this was a really nice diagram for y'all because it shows the molecule and it shows what happens as you go through. Okay, so we're going to take it step by step. But what you have to remember when these reactions are occurring, oops, I didn't want to do that. I want to make it smaller. There we go. When these reactions are occurring, they don't all have the same free energy. So if you have a really high negative delta G, right? Like here, negative 16.7, negative 14.2, negative 31.4. What can we say about these reactions that are in blue? They're energetically favorable, they're spontaneous. And so what that means basically in the cell is that these reactions, these three enzymatic reactions, are irreversible. Because they have such large negative delta G values, right? 1, 2, 3, these are irreversible. If you have something that has a delta G that's really close to 1, and really we're looking in this column because this is what happens in the cell. If you have a delta G value of negative 0.4, you could really go either way. So basically those are reversible reactions. But these are all irreversible reactions so that the pathway will continue to progress forward, right? And so we're going to look at each of these and see why they are irreversible. So our very first one, right? We're going to start with hexakinase. All right, hexakinase. This is we're in part 1, right? Part 1 is the investment phase. Investment phase, right? And we really have two big goals inside of our investment phase, right? We want to use ATP. This is our investment. We're spending ATP. But what are we going to use ATP for? Like, why do we have to invest energy? We're trying to produce energy. Well, it's pretty close. It's pretty close. So we want to use ATP to transfer. It's going to be the donor, right? In the donor. So we're going to transfer a phosphoryl group. We're going to make phosphorylated compounds. Why do we want to make phosphorylated compounds? What did we learn about phosphorylation events on proteins? How did we mimic? That was one of the questions, the questions last chapter. It was a mimetic, right? We could mutate this group that could be phosphorylated into something, and it would behave like it was phosphorylated. What was the property? A what? I can't hear you. Well, yeah, so you wanted to mutate that protein, right, to make it basically always phosphorylated and always on. So what kind of residue change was that? You wanted to mutate that residue that could be phosphorylated into a residue with a negative side chain. Remember this? Maybe, sort of. You have to pull. We're pulling from last time, right? So what we're trying to do is we're trying to ensure that glucose, how does glucose get into the cell? But like how does it get in? You can have a transporter. So once you get it in, what keeps it from leaving? What are you going to do when you add a phosphate group? You're going to make it negative. You think it's going the same way it came in? It can't. It's negative. The whole point of phosphorylating something, adding a phosphate group onto a sugar in glycolysis, is to make sure that it can't leave the cell. You're trapping it in the cell, right? It's kind of cool. And then, so that's one thing, you want to trap it. So to trap in cell, so that's one, use ATP, right? Phosphorylate it, trap it in the cell. And then two, the second thing is we want to break it, right? We want to make it into, turn it into 2 3-carbon molecules. And so that's what we're going to see in that investment phase. Those are the two things that are going to require energy. We need to trap it, and we need to turn it into 2 3-carbon molecules. So our first step in the investment phase is hexakinase. And if you remember, hexakinase was one of those irreversible steps. There are only 3. You need to know them. There are only 3. You need to know them. So there are actually 2 different enzymes that catalyze this first reaction depending on what cell type you're in. Either it's hexokinase, which is like most of everything, or it's glucokinase. And glucokinase does the same thing, except that it is inside of your liver and it's inside your pancreas. What does your pancreas regulate in terms of sugar? Insulin. So we're going to see, I'm going to show you in a minute why that's really, really important and why glucokinase isn't quite exactly like hexokinase and why that's important. All right, so this works in an induced fit mechanism, right? And your book goes into way detail on exactly how those enzyme kinetics work. You don't have to go quite that deep. You don't. All right, so this is a phosphorylation reaction. So we start with our glucose, and what are we going to do? We're going to add a phosphate group to it. So what kind of an enzyme is it? It's a kinase. Yes, it's a kinase. This makes sense. Yay, we're finally naming things well. All right, so it's going to require, where did that phosphate group come from? Came from ATP, right? Now, what's interesting about all kinase reactions, is that the ATP substrate for this reaction is actually in a complex with a divalent cation, Mg2+. And if you don't have that Mg2+, this reaction is not going to go. So what do we know about a phosphate? Look at the charges. Negative what? -2. What's the charge on magnesium? +2. Kind of makes sense, right? So it's to shield those negative charges in order to allow these reactions to go. So if you don't have magnesium 2+ there, the reaction's not going to go, right? Okay, so this is our first step. We're just adding a phosphate to our glucose, and we're going to make glucose 6-phosphate. What carbon does that phosphate add to? Carbon 6. Okay, now we said there were 2 enzymes that could do this. Now, your body uses not hexokinase but glucokinase to monitor your blood sugar, right? I know we say if you're diabetic, you're going to use a glucose monitor, right? Well, you have an inherent glucose monitor inside of your body, and if you are functioning normally, it's working. So this is our artificial, right? But what happens in the body? What's the real deal? Here's the real deal. In normal cells, you have hexokinase, and in your beta cells of your pancreas, you have glucokinase, right? So that's beta cells of pancreas. What do you notice about the affinity of glucokinase versus hexokinase for glucose? Which one has a higher affinity? Hexokinase. So in order to activate glucokinase, you're going to have to eat a lot. Oh, I don't know, maybe the size of a meal, right? Then that's going to increase your blood sugar high enough that you're going to start activating glucokinase, right? You don't want glucokinase activating and releasing insulin and activating this cascade mechanism that I'll talk about in a future video. When you only eat a little bit, right? Then that would tell all the cells of your body to pick up glucose from your bloodstream, but really your blood glucose levels are low. So do you want to pull glucose from your bloodstream when your blood glucose is low? No, you're going to pass out, right? Very quickly. So what you want to do is you want to wait until you've eaten a meal. Once you have a meal and you have a high enough blood glucose level, then you're going to activate glucokinase and initiate that cascading system to release insulin. There are different places within the pathway that will lead to diabetes. And so you could have mutations in a couple of different places.
Okay, so it doesn't vary on the insulin with the pancreas. Like if it did, like if we're resistant to insulin, like if we're
So if your cells are resistant to insulin, that's something else. You're releasing the insulin, right? It's going out into your bloodstream, but now your cells in your body are like, what? Even though you've released insulin, right? So that's your different types of diabetes. Either you're not producing insulin, or you have a mutation in the insulin gene itself, and so the insulin made is defective, right? So that's kind of two different things that result in the same thing.
So would it alter the glucokinase?
You can have a mutation in glucokinase, and that would definitely affect and give you a form of diabetes. Yeah. So, but there are a lot of different ways. Oh, we are almost done. Yes, I'm going to record the rest of 9.3. Well, what I'd like to do is try to go over the rest of 9.3 in class on Wednesday, and if we get to a couple of the discussion questions, that's great. But please do know that there is a hurricane in the Gulf, and I don't know how that's going to work. So what I will probably do is make a video today of the end of 9.3 in case something happens. And that way before power goes out and all that kind of stuff. And then I will message you on the announcement board as soon as I know what's going to happen, but it's supposed to make landfall on Wednesday. So I will make a recording for the end of 9.3. So let me put it that way. And then we'll make a decision from there about what's going to happen. Okay. So I'll try to go make that later today, so you have it by like 5 o'clock today. Okay. All right. Any other questions? But my watch is early. We still got a few minutes, right? We got a few minutes. I want to show you the next. I really want to get through the next irreversible one, right? Do we have time? Oh, it's 11:40. Oh, okay. Yeah. Go ahead. Sorry, y'all. I'm just so excited about glycolysis. All right. So y'all have a good rest of your Monday. Hopefully the hurricane will not come our way. If you have any questions, send me a message.
So the whole data thing.
Yes.
Whenever you go from data, flip the whole thing.
Uh-huh.